it's live. Okay. So I will touch on some of the exciting things going on in healthcare in general. I will not have time to talk about regenerative medicine, which in any case, people smarter than me have already discussed. So let's start with something that has to do with neuroscience, leading towards something that I call the brain on a chip. The challenge of neuroscience, there are two challenges, temporal and spatial, time and space. In time, you have this paradox that a single neuron operation is at milliseconds. But of course, changes in your brain can take months or years. In space, you have the problem that single neurons are very teeny, 10, 100 micrometers, but the neural regions, the brain regions that you measure, if you unfold them, are several meters long. It's a strange science, and it is having problems becoming a technology. In fact, the success of neuro drugs is among the lowest of all in medicine. There are new tools that have been very important. I will mention only some. At Stanford in 2005, introduced optogenetics for monitoring and controlling neural activity. Basically, you add some DNA to a neuron's DNA so that the neuron makes proteins that they call opsins that respond to light. And the famous experiment was when the rabbit was emitting blue light. Optogenetics is now being used for different functions, and so it's very real. It's no longer just a novelty. Also, I will mention this experiment, which is sort of typical of what is happening. It used to be really primitive, the way we were measuring, detecting neural activity. A few years ago, 3 labs at Stanford got together and they built this device that can record the activity of hundreds of neurons at the same time. Basically, it makes a movie of brain activity, and it connects directly to silicon for applications like brain-to-machine interface. And the same GLU at Harvard has made a recording of neurons over the whole adult life of mice. So lots of progress in the fundamental tools. Now there are platforms to make neurons by using IAPC-induced pluripotent stem cells, the great invention of 2006. And these platforms make neurons that you can use for what I call brains on a chip. You can create your own brain. In fact, this startup in Australia, Cortical Labs, creates these quote-unquote chips, which are a mixture of silicon and tissue. And so they claim the chip is alive. And of course, some people speculate that maybe this would be a faster way to get to general AI. Few words about brain organoids. By growing neurons, you can put them together and create biocomputers. So in a sense, you're trying not so much to recreate the human brain in vitro, but recreate aspects of the brain and not necessarily the human brain. This means that we don't need to perform horrible surgeries on animals. And And so these are some of the people working on brain organoids. And I will mention mainly Alison Muotri, UC San Diego. On YouTube, you can find a conversation I had recently with him, which is very, very futuristic, but also very promising. A few words about Xenobots. These are computer designed organisms. The cells are real cells. They come from frog stem, from frogs, and they're genetically unmodified. They're really the cells of the frogs. And then these organisms are created, and the computer uses an evolutionary algorithm to decide which ones have to survive and to make random copies of the survivors. And so I mentioned down below the scientists working on this project. A few words about biosensors. These are so that there's a lot of startups working on biosensors. They would be very important to improve health and for precision medicine. I'll mention Jennifer Dion at Stanford. She's working on silicon photonics to enable very fast reads of biological information. And Michael Snyder is creating wearables that put together a number of different technologies. Something about RNA technology. It dates back from the 90s. One of the pioneers was Catalina Carico at University of Pennsylvania. And in the 90s, the first mRNA vaccine was designed, and but then Catalina Carico and Drew Weissman showed how to make mRNA vaccines. And a few years later, two Turkish immigrants in Germany founded BioNTech, while Moderna was born when Derek Rossi discovered a method to make gene therapy based on mRNA. So messenger RNA has a long story, actually. And BioNTech developed a cancer vaccine based on Carico's method. As you all know, in 2020, Moderna and BioNTech used that technology to create the COVID vaccines. The technology was there, luckily it was there just when we needed it. Now, in theory, we can make messenger RNA for pretty much any protein. And so potentially we can target any infectious disease and in relatively rapid time. That's the beauty of messenger RNA, is that it's easy to edit. So potentially this kind of vaccines can encode more than one viral protein. And this was proven last year by Norman Party. Then the dream, of course, is a vaccine on demand, which becomes more realistic with this technology, or at least a vaccine that works on multiple viruses. Just think of the fact that the flu vaccine has to be remade every year because the virus mutates so quickly. So the dream is that someday we could have a custom-made vaccine therapeutic to treat even cancer. You study a person's tumor, and then you can use a messenger RNA to create a custom-made treatment. So that this is not science fiction. There's a test trial underway by BioNTech and Genentech. Cancer vaccines are tricky. We'll see some later because it's not clear which protein you have to target. And the same technology is being used for other disorders like cystic fibrosis and sickle cell and anemia. And just a few months ago, Vertex announced some promising results. During COVID, there's been a lot of talk about antibodies, just transferring antibodies from one person to another. And that's the convalescent plasma technique. The problem is that we know very little about antibodies, so we have to study a lot more about antibodies. And so I mentioned Chris Barnes at Stanford, who is classifying the antibodies, and then you can start thinking what would work, which one would work on coronaviruses. Now, immune therapy, of course, is an old idea. There's T cells that are immune cells that identify other cells infected by a virus or cancer. And CAR T cells are genetically engineered T cells to fight cancer. 6 years ago, we had the first T cell therapy approved by the FDA. And in 2017, there's been a boom of startups in this field. Finally, we have some success stories to share. The first one I mentioned is Kite's Tecartus, which has good response in a test. And then I mentioned the UC Davis started a clinical trial to use CAR T cells to cure HIV. And FDA so far has approved the 6 CAR T cell therapies. I'm aware the CAR T cell therapies are not the only immunological therapy available, and I will only mention T cell receptor that makes T cells. Now back to cancer vaccines. There are basically 3 kinds: cell-based, protein-based, and gene-based. In 2010, this company, Dendreon, introduced a prostate cancer vaccine that was the first ever cancer vaccine approved by the FDA. And as I mentioned, BioNTech was working on a cancer vaccine. Now, COVID accelerated messenger RNA technology, and that will probably benefit also cancer vaccines. So we have something from Moderna, we have something from BioVax, and so on. So that's multiple companies are working on cancer vaccines. Different ideas on how to do it. Now, a little bit about therapeutics and then a little bit about diagnostics. In therapeutics, a major, if not a revolution, a major change would be if we could have ultra long acting therapeutics. Therapeutics medicines that last whose effect lasts a very long time. And Robert Langer at MIT has been working on this, and now that is being commercialized, and we are beginning to see the emergence of startups in this field. Diagnostics, liquid biopsy has been around for a while. There was a boom, especially in the Bay Area, but throughout the United States, of liquid biopsy startups, some of the best funded biotech startups at the time. Grail is by far the one that got the most money, and it has delivered a Gallery test that is a multi-cancer early detection test that can work for more than 50 cancer types. So the first promising results were just announced. It was a trial in England. And the good news, as you can see in the BBC article, it shows promise. The bad news, you can see it in the Reuters article, Grail admitted that about 400 patients were told that they had cancer, but they didn't. So it's still not perfect. This is becoming popular. The health selfie, you take a picture and an app tells you something about your health. Now, AI has been applied especially in image recognition, in taking a look at a medical image and understanding something about the patients. It has also been used for scientific research. AlphaFold is the notable event in which AI contributed to a major scientific discovery. And the big promise is to use AI for drug discovery, especially to accelerate drug discovery, picking the right target in the body, designing the right molecule to interact with it, and deciding which patients are more likely to benefit from this. And so here I mentioned some of the startups. This was interesting. I mentioned it because it was a little different. At McMaster University, they built an AI model trained on drugs that we know, and then the system was asked to compile a list of potential antibiotics, and in just a short time, it helped scientists discover a new antibiotic, Biosensors, which at the time was considered promising for COVID. Then I don't know exactly what that happened. And of course, the promise of AI is that once you have big data, biotech, wearables, robots, we can build smart hospitals. And there's all sorts of questions and about how we train nurses and physicians, and how the patient will interact with the healthcare system. And we have some significant tests going on in hospitals in the United States. Precision medicine has been around for a while. There was a national program in the United States, and these are different startups that provide the platforms for precision medicine in different parts of the world. A few words about gene editing. CRISPR is more than 10 years old by now. And you probably remember 2016 when there was a boom of CRISPR startups. What happened since then? Well, just a few days ago, the FDA approved something that was developed at Washington State University. These are CRISPR-edited pigs, and the FDA approved that humans can eat them. So, sausages will be made out of these CRISPR-edited animals. And last year, Alex Marson, who leads a big team at UC San Francisco in collaboration with other institutes, developed a new variation of CRISPR that makes it easier to re-engineer massive quantities of cells. And this would be useful to create a new medicine. For example, they generated 1 billion CAR T cells that target multiple myeloma. All of this relies on sequencing machines. There's this machines called next generation sequencing, then originally introduced in 2004, and then Illumina became the leader, and now we have some that really cheap. But many rare diseases are not diagnosed because of the short read methods that these machines employ. So the third generation is machines that are capable of long reads. And I mentioned Pacific Bioscience and Oxford Nanopore that makes the introduced years ago a portable sequencer, and now it's collaborating with NVIDIA to accelerate its sequencing, the sequencing of what it reads. Few words about cell-free systems. Most of synthetic biology today relies on cell-based system. The scientist takes a living cell and then introduces some new genetic instructions in it. That works, but we don't have cheap biosensors, because that technology doesn't lend itself to make cheap biosensors. And biosensors would be, as I mentioned before, would be important for measuring, monitoring, and so on, our health at the end of the day. So that's in vivo. Mostly synthetic biology today is in vivo. Cell-free systems are in vitro. They don't use a living cell. They use biochemical reactions to create something that behaves like a cell, but it is not a cell. It has the desired genetic instructions. This is an old idea. In 1961, these scientists are the ones that discovered how to decipher the genetic code. Well, they use cell-free systems, and also the most widely used cell-free technology, CFPS, also dates from even before 1961. In 2017, a Japanese scientist showed how they could use cell-free systems to develop biosensors. And that sort of revived the whole field. Now I will mention this team at Northwestern University and Shanghai Tech University. They used a cell-free method to make valinomich. They increased production by more than 5,000 times. So now there are a number of these platforms. Now, what is this artificial cell? The artificial cell, basically, it's a programmable liquid that turns DNA into RNA into protein. So just like a regular cell, but it is not a living cell. They consider it safer inside the human body, and it's promising because it makes it easier to manufacture biologics, and they can also be stored at room temperature. So the dream is that they will allow us to manufacture vaccines and biosensors rapidly in large quantities. In theory, everything can be done in cell-free systems. And so this would be a revolution similar to the revolution in computing caused by personal computers. All of this is made possible by equipment that I'm not discussing. So we have to pay tribute to the new machines. The ideas are old. Of course, there's still a lot of work to do. Meanwhile, I want to mention, since I was talking about the central dogma of biology, genes, messenger RNA to protein, I want to mention that in the last few years, there are doubts about the dogma. Anthony Jose at the University of Maryland, for example, he noticed that the DNA does not contain all the information. For example, the DNA doesn't say where the eye will be. Some crucial information must be somewhere else. And he thinks there is no linear DNA, RNA, protein flow, but there is a cycle of cycles. And we already know for more than 50 years about the transcriptase which synthesizes DNA from RNA, and the genetic mosaicism is widespread. In 1976, a Japanese scientist discovered that the cells of the immune system contain slightly different genomes. And that's actually useful because it produces antibody diversity. And in 2001, Gerald Chun shows that not all neurons are genetically alike. If you go on YouTube, you can find a video of my conversation with him. So every person contains a multitude of genomes, which is not the dogma, the original dogma. Now, all of these things, you put them together and you get to the most exciting part of biotech. I call it the science of longevity. Why is the Hydra immortal? Why the Turritopsis can reverse its life cycle and get younger? And why did some people live to such an old age? What's special about these people and these animals? So, 1999, two distinguished scientists, Lenny Guarente and Cynthia Kenyon, started the Elixir, and I see that as the beginning of these startups that want to extend the human life and ultimately cure death. In 2004, David Sinclair started Sirtris, which still exists. In 2013, Google started Calico, which still exists. Craig Venter is a famous biologist. He started human longevity. Craig Venter is one of the people who sequenced the human genome. In 2020, David Sinclair at Harvard posted a major encouraging result. He showed the mice that regained eyesight. Coincidence or not, a few months later, there was in Switzerland, there was the first conference of longevity investors who put money in this kind of research. And last year, there was an important conference, the Gordon Research Conference in Maine, that put together all these names. These are all famous names in this field. Now, I call them reprogramming startups, non-longevity startups. Why reprogramming? Because they aim at reprogramming our genes so we can live longer. And look who founded them. Retro Biosciences got money by Sam Altman, more famous as CEO of OpenAI, billionaire. New Limit was started by a billionaire, Brian Armstrong. Cellularity was again started by a billionaire, Peter Diamandis. Altos Labs was started by two billionaires, Jeff Bezos of Amazon and Yuri Milner, a Russian. Biosplice was founded by a billionaire, Osman Kiber. Juvenescence in Ireland was founded by a billionaire. So there you have it. And there's many more, of course. And then there's platforms that claim to enhance longevity. Somebody counted more than 300,000 health apps. I would mention only telehealth, founded by David Sinclair, who I mentioned before. The problem with reprogramming, cells lose their original identity, right? You reprogram them. So somebody at the Babraham Institute came up with the idea of transient reprogramming, so that that reprogramming happens only for a short period of time, and you still get the benefits. Now, how do you measure if this science is working? And so we discuss aging clocks. That's a way to quantify aging, and it's also an indicator of overall health. Indirectly, should be a predictor of longevity. Unfortunately, the original clocks were chronological, and they were good at estimating your real age. What we need is biological age, and many factors contribute to aging. So what you need is really a combination of biomarkers, such as telomere shortening, genetic instability, epigenetic alteration, accumulation of misfolded proteins. You have to measure all these things. So let's look at the epigenetic clocks. They're based on changes over time in DNA methylation. I mentioned two that opened this field. And the second generation especially designed to measure biological age. And I mentioned a few. You can read the scientists involved in this research. Then there's this kind of aging clocks, clocks that read the biological age of an organism directly from its gene expression. And so I mentioned two of the scientists who are working on this. And of course, somebody's already using deep learning. This is the founder in silico. As far as I know, it's the first such aging clock. The truth is that different cell types may have different aging. You may be getting deaf, but not blind. So those are different cell types. And so somebody came up with the idea of measuring different clocks. Okay, these are some of the commercial products. There's actually a lot. Bottom line, there's an increasing consensus that biological aging can be reversed. It is possible to make people younger. And we have powerful tools now. We have powerful tools to identify if therapies work. There's a few foundations and institutes that work on this. I will mention young blood because that's the famous therapy that the media advertised, especially the 2014 experiment where young blood seemed to restore the faculties of all brains. Unfortunately, a study in 2019 showed no benefits. Nonetheless, in the last 2 years, there have been new announcements that have to do with the young blood. So maybe there is something to it after all. Another promising approach is through MSCs that are harvested from these are cells that are harvested from the bone marrow of adults. And they seem to have done well in a bunch of different areas. That's Gary Steinberg at Stanford working on this. And then you rediscover what our grandparents already knew: exercise. And the benefits of exercise are well documented by now. These are some of the things that you get if you exercise. Your brain even generates new neurons. Okay,